Okay, so welcome everyone. Thanks for joining us. My name is Amy Franco and I am the Assistant Director of the Adult Department at the Glen Ellyn Public Library. Welcome to the COVID Q&A with our very special guest, biomedical engineering researcher, Rebecca Keate. Rebecca is a PhD student at Northwestern University, and I'm also very proud to say that she's a Glen Ellyn resident and is generously volunteering her time and expertise to answer any questions you have about COVID-19. Rebecca researches electrical conductive biomaterials for tissue engineering and holds a Bachelor of Science in Biomedical Engineering from Georgia Institute of Technology. Her research includes bacterial infection, cellular mechanisms of cancer, and tissue mechanics. I will be moderating this conversation with Rebecca and asking questions on your behalf. If you have a question for Rebecca, please type it into the chat. If you have any problems with Zoom, we recommend exiting the meeting and rejoining with the same link or meeting ID. If this is your first time using Zoom, you may need to adjust your settings and then restart the application. If you still have issues after restarting Zoom, please type into the chat and a GEPL staff member will assist you. So with that, I will turn it over to Rebecca. Welcome, Rebecca.
Thank you so much, Amy. So like Amy mentioned, this is meant to be an interactive presentation, and I've gone through a lot of common questions that I've been asked about COVID-19 and compiled them into a presentation. So if at any point something is particularly interesting or confusing, please don't hesitate to send Amy a question in the chat. So I would like to start today with just a little bit about me. So I have a Bachelor's of Science from Georgia Tech, and I'm currently a PhD student doing research at Northwestern. So my goal in this presentation is to kind of serve as a liaison between what we hear about the research that's going on related to COVID-19, since that's a lot of the progress that's currently happening is in the academic and industrial labs, and translate that into what that actually means. A lot of us don't actually have direct interactions with research and don't know what all the steps are or what it necessarily even entails. So I'm aiming to answer some questions and alleviate some of the black box about what a lab is actually like and what biomedical progress, how it actually happens. So I have industrial experience at Johnson & Johnson and also Becton Dickinson, both in medical device development and in the research design field. So with that being said, I would like to jump into a little bit on where the virus came from. So there's a lot of confusion about, it seems like this kind of appeared out of the blue. So just how do viruses infect humans? The way this started, what we think so far, is that it originated in a bat. And the way we can make that conclusion is because about 89% of the virus's genetic material, now that includes things like DNA and RNA, resembles that of Chinese bats. So the current hypothesis is that this virus was native to bats, transferred to another animal, some sort of an intermediate host, where it mutated and then evolved the capabilities to infect humans. Now, looking at this triangle diagram that I have on the slide, it might seem quite straightforward in how this process happens, but in actuality, it's very complicated, and it's sort of like the perfect storm that happens in order for that mutation to occur. There's a lot of very specific changes to different proteins and their molecular structure that happen in order to facilitate that, that infection from bats into another animal, such as a pig or a mouse, and then go into a human. So that's how a lot of these new infections actually come to start. And that's because when animals are in really close proximity to each other, there's a lot more interaction or opportunity for infection from one animal, one species, such as a bat, into another, and then into a human. So it's really rare that this happens, and it's a lot of random effects have to occur. So this information came from the CDC. There's been a lot of concern about how to protect yourself from the virus. The good news is that the virus is very easily removed from surfaces with disinfectants. The types of disinfectants that you should be using can be anything like bleach when it's diluted according to the manufacturer's instructions. One of the biggest mistakes that individuals might be making is using expired disinfectants, or also not cleaning off a surface before actually disinfecting it, because then you might not actually be getting all the viruses or all the places that the virus could be. In addition to that, avoid touching your face. If the virus is on your hand and you haven't washed your hands, if you touch your nose or your mouth, that gives the virus basically a route to your lungs where it becomes potentially problematic. With that being said, wash your hands often. If those resources aren't available, make sure that hand sanitizer is at least 60% alcohol. And then in addition to that, avoiding contact with individuals that might have the virus is one of the safest ways to protect yourself from transmission. I also want to talk about the cloth face coverings, since there's been some misconceptions from people that I've talked to. The number one thing that cloth face coverings are beneficial for is preventing transmission from asymptomatic individuals to the rest of the public. One thing that makes COVID-19 quite unique is that about 40% of people that are carrying the virus don't have any symptoms whatsoever. This is not typical for viruses, and that's why the cloth face covering helps reduce the airborne transmission of particles from your nose or mouth if you're not aware that you're actually infected. So you shouldn't wear the face covering if you have trouble breathing, are unable to remove the mask without assistance, and they're also not recommended for children under the age of 2. Since face coverings are now required in Illinois and recommended in many other places, or they will be required quite soon, I've included this diagram on the slide that I got from the CDC website that I think is really beneficial. Personally, I don't have a lot of experience with sewing, so looking at this diagram of how to craft a mask from a t-shirt is really important, because anyone can make these masks even without any sewing experience. So keep in mind that a double layer of fabric is recommended to truly prevent the transmission of the virus particles from your nose or mouth. And so you can cut out the shape of the mask and then tie the t-shirt in the shape of strings to secure it onto your face. Now I want to talk a little bit about testing. So there's been a lot of uncertainty about how testing works and why it's so important. As I mentioned, a lot of individuals are asymptomatic. The way that we are currently working to diagnose people that are infected with coronavirus is by using a method called reverse transcription quantitative polymerase chain reaction, or just RT-qPCR, or it's also very commonly referred to as just PCR. And this is the main testing approach that we currently have. So it's the gold standard for identifying the infection of the virus. The way this method works is there are specific codes in our DNA, and there are also specific codes in the virus DNA. So during this method, we have, if you look at this diagram, these little pink sections represent a sort of Lego block that we are attempting to match to a certain code of the virus DNA. So if this pink Lego block is able to match a specific sequence, then we know that after collecting a sample from a person, that there is a fragment of the virus DNA intermixed with the human's DNA. So PCR separates the strands of your DNA, since it naturally exists in a double helix form, like a sort of ladder. So during PCR, these strands separate, and then a single strand is searched for this Lego block insertion. And once the Lego block insertion clicks into the single strand of DNA, it's extended and a new double strand of DNA is formed. And this is considered one cycle of qPCR. So this method is quite complicated. And if we think about all the steps that are required, this is quite time consuming and expensive. It requires a lot of specific equipment. It requires certain certifications for labs to actually perform this test. And it also requires some very specific chemicals. So if you think about actually matching a sequence of DNA, it's quite expensive to develop that sequence and make the match yourself. And also, you have to go through cycles of repeatedly trying to find a match, elongating the strand, and then measuring the amount of matches that you get in that solution. So I know this method is quite complicated, so I'll pause here if there's any questions or confusion about what exactly is going on when we're trying to detect the virus DNA.
I haven't seen any questions yet about this process, but we do have a couple of questions about masks and filters, if I can ask those. So Maria would like to know about if your mask should have a filter, and if so, what materials are the best for a filter for the mask? And I'd also like to know, if you are going to make your own mask, what kind of materials are better? Do you want to make it out of a cotton t-shirt? Do you want to make it out of a polyester, like a gym shirt? Can you talk a little bit about that?
Yeah, so in terms of a filter, I'm not sure that that's entirely necessary. Like I mentioned, the main objective of the masks is to prevent bulk transport of particles from your mouth or your nose into the air. So unless you are going to be, for healthcare professionals, the filters are much more beneficial. And I'll talk about the difference between healthcare professional masks and masks for the public a little bit later, a little bit more. But the filters are primarily to more so protect individuals from getting the from picking up the coronavirus. So I think if you're in the public, that might not be as necessary because you're at a much lower risk, and it's primarily just to stop the transmission. In terms of materials to make the mask out of, really the best materials will be the materials that stop airflow or liquid particles the best. One way that you can actually measure this is if you have a spray bottle, you can fill it with water and you can spray through the material, and a lot of iPhones or other smartphones have the capabilities to take videos or take slow motion videos. So if you're trying to figure out what material is best, you can spray water through the material and then take a video and look back at how much water actually gets through the material. And the idea is to have something thick, like I'm guessing cotton would be one of the better materials that will actually absorb those particles.
Interesting. And so we have another question. Should we be covering our hair to protect hair particles from COVID in the air?
Yeah, I have not seen anything so far that hair is at risk. I think that I've seen a lot where the virus particles, they don't think it can actually live on your hair. So that shouldn't be too much of a concern, but I would still exercise caution with touching your face or touching your eyes or your nose or any orifice where the virus can be transmitted into your lungs. I think the hair is a lot less of an issue.
Okay, so I think, unless anybody has any other questions, I think we can continue.
Awesome. Okay, so this is just a little bit about testing. So moving on. I want to talk a little bit about social distancing. So just in general, why this is something that's important and why we're making a large effort to actually social distance. So the first thing that I want to introduce is this R naught, which is called the reproductive number. And it refers to how many people on average one person with the virus will infect. Now, when COVID was first introduced, it was predicted that the R naught would be about 3, meaning that each person who was infected with the virus would infect 3 other people. So I would like to introduce this diagram just to think about for a second, because it shows how the effects of social distancing can really save lives. Where the virus first started in Wuhan, China, they were the epicenter of this entire outbreak. So they really had no way to even begin to prepare for the virus or stop its transmission. So not only did significantly more people become infected than even in the rest of China, about 50,000 people in Wuhan were infected compared to about 15,000 in the rest of China. But we also see over 5% increase in fatality rate. So what that means is that when hospitals become overrun or overwhelmed and resource limited, a lot more people are at risk for dying because the hospitals don't have the resources to treat everybody. So using this as a model, since China tackled this virus before it got here, we started to think about social distancing to prevent such a prolific spread like Wuhan had, and then also to stop our hospitals from being overwhelmed so we can truly manage this virus. So in the bottom, you'll see a graph with different projections of the number of cases over time. So the first red line shows the biggest increase in the number of cases over the shortest amount of time. And what that's describing is how many people would be infected with no social distancing measures. And you see that the outbreak is a lot, it happens over a shorter period of time, but a lot more people become infected, and then we're at risk for significantly higher mortality rates like we saw in Wuhan. Now, when we implement social distancing measures, that is represented by both the blue and the green lines, you can see a significant decrease in the number of people being infected. However, what the blue line represents is when social distancing measures are removed too soon, you no longer get that smooth curve, but you actually get a resurgence of infection. So the moral of this is there are many factors that we don't yet understand in terms of how to figure out when it's safe to stop social distancing. What we're trying to prevent is a resurgence of the infection, like that what could happen if you remove social distancing too soon. Now, also considering the effects of social distancing, it's very difficult to make these projections because of the changes to the reproductive number, which is how a lot of the predictions are made in terms of where the peak of the virus will come. As I originally mentioned, the R naught was predicted to be about 3 for COVID-19, but with social distancing, it's around 1 or 1.6, which makes it very manageable just over a longer period of time. I can pause here. I see that there are some questions popping up.
We do have a few questions. We've got one wondering how long it lives on different surfaces and whether it can be transmitted through the air.
Yeah, okay, so it can be transmitted through the air, and that's a lot of what the masks are working against. Wearing the mask, the aim is we can all stop it from transmitting ourselves into the air. And then in terms of how long it lives on surfaces, there's still a lot of investigation going on about that. it's hypothesized right now that it can live on tough, harder surfaces for hours or days. And then softer surfaces, it doesn't seem to do as well on. But as long as you are disinfecting, it won't actually survive. But also keep in mind that there's still a lot that we don't know. There's been very few studies that have actually occurred in places like hospitals about how long the viruses live on surfaces. And you have to keep in mind that there's a much higher concentration of the virus there. So it's easier to see it on surfaces. In terms of how long can it live on a surface inside our own private homes, that's really not well characterized, but it seems like it's much less of a threat compared to airborne transmission, or if it's on your hands and then you touch your face or your nose or your mouth or your eyes.
So can you tell us if, there's a lot of talk about airborne transmission and that's why we're wearing masks. Does it stay in the air? Is it, tied to like a sneeze or respiration or something like that?
Yeah, so it's usually tied to a sneeze or respiration, but virus particles are traveling with the saliva or the liquid, the air particles from your mouth or your nose. So they don't necessarily linger in the air longer than your actual saliva or nose particles. I don't actually know the timeline of how long they linger in the air, but they will eventually fall to the ground, just like any other drops of liquid or aerosol.
Okay, and so in terms of routes of transmission, I know that we're not supposed to be touching our eyes, nose, mouth, anything that could potentially get it into our lungs. But do we know if it's worse to touch your eye, for example, or is it worse to touch your nose? Can you talk a little bit about routes of transmission?
Yeah, I think that's still quite unclear. I think that there's none that's identified as worse or better than another. The more direct it is to your lungs, I'm guessing would be the worst. But there's a lot of new studies coming out about the eyes being dangerous. So I don't think that means that the eyes are better or worse to touch than your nose or mouth. I think it's just best to avoid touching your face in general. I think that a lot of researchers it's very hard to isolate if someone has contracted a virus specifically from touching their eyes or touching their mouth or touching their nose. So it's not clear yet what's the most transmissible method.
And so in terms of social distancing, when we do have to go out into the community, we're hearing a lot about, stay 6 feet away, wear a mask, but there was recently a study done with, like, I'm sure you saw this, the glycerin smoke and the laser and like the artificial sneeze. And they were finding that the sneeze can be transmitted much farther than 6 feet. So why was it that 6 feet was chosen and not something more distant, like 8 or 10 feet?
I think that 6 feet was the safest guess. I think that a lot of politicians right now are just trying to make the best decisions. And I think on average, 6 feet is probably safe to protect yourself from a sneeze. Sneezes are quite different depending on person to person. So I think 6 feet is just the average distance that it's the safest, and you're not necessarily breathing in the same air from each other, especially with a mask. That definitely stops the distance of transmission.
Okay. And I know in this slide you were talking about, the reason that the fatality rate was higher in some areas might have been because of, hospitals becoming overwhelmed and, the public health infrastructure. But from a public health infrastructure in this country, from a public health perspective, I should say, can you talk about health inequities and why the fatality rate may be different in some areas of Chicago than it is, in DuPage County, and how kind of the politics of health equity plays into place here?
Yeah, that's a really quite complex question. And I think the process has started. There's been a lot of different factors that have kind of played into each other. The first is distribution of information. Different areas of Chicago necessarily, I saw a lot of studies weren't as aware of the severity of the virus. And that's one reason why it started so potently in those certain areas. And then being having a less of an understanding about the severity definitely affected the seriousness of social distancing and how seriously people followed those guidelines in those areas, which propagated the spread. Now, once you already have propagated spread, and we know that there's a higher chance of fatality rate, and statistically, when more people contract the disease, there's going to be more negative outcomes. So I know there's a lot of studies that are going on right now trying to understand the health equities of the health inequities. And I've seen so far, the one thing that they are sure of is that it started with lack of information and not as comprehensive of an understanding of the virus in general. And then there's also different resources. As I mentioned earlier, things like washing your hands, certain populations might not have as easy access to as effective of soap or as effective of disinfectants or disinfecting wipes, which also limits them in their ability to clean their homes or clean their hands and stop the spread. So I think it's mostly related to the transmission of the disease being higher in those cases.
Okay, and then when we do, back to social distancing, when we do have to go out, I mean, everybody's got to eat, everybody's got to go out and get their, medication and things like that. But when you come back or you order takeout or whatever, do you really need to be wiping down your groceries? Do you need to, not touch your medication for a few days? What's safe?
Yeah, I've seen a lot of professionals say that you don't need to be wiping down your groceries. However, if it makes you feel safer and it makes you more at peace and less stressed, then it's definitely beneficial. Because another thing to keep in mind is how your state of mind, that's important to maintain during such stressful times. So if you're worried about contracting the virus from your groceries, then by all means, wipe them down if it makes you feel better. At the same time, you don't necessarily need to be worried about contracting the virus from your groceries, especially since a lot of places themselves are taking a lot of precautions and doing advanced cleanings, and they have shortened hours to be doing these thorough disinfectants, and that all the employees are typically wearing gloves and masks. But yes, like I said, if it makes you feel better, then that's definitely something, an action that you can take. And I think that periodically wiping down the surfaces and disinfecting the areas in your house is a best practice.
Okay, and then one more question before we move on. Let's say you are able to live completely in a bubble in your home. Maybe you grow your own vegetables, you're not going out, your risk of exposure is quite low. Is it still necessary to disinfect your home? And where's the line in all of this? If you're really not going out into the community, you're not bringing things in, you've got all of your own stuff, everybody's staying at home, what do you really need to be doing to protect yourself at home?
In that case, it's not, if you're quite isolated, it's not necessary to be disinfecting. I don't think you only have a way of contracting the virus if you contract it from someone or you touch something that's contaminated. So if the virus doesn't have a way to get in, you don't need to be concerned. Like if you're going out regularly, then it's definitely best practice to be disinfecting surfaces. As you mentioned earlier, Amy, things like takeout or groceries. It'll just keep you safer if you're regularly disinfecting, but if you're isolated in a bubble, then you'll be fine. And there's almost definitely no way for the virus to get in unless it's got some route.
Great, thank you. I think we are ready to move on.
Awesome. Okay, so just a little bit more about social distancing. There's been a lot of questions about how long will the stay-at-home order last? And the easy answer is that there's a lot of mathematical projections that are being used to estimate the peak dates, just as was done in this graph below here. And the real answer is we're looking for 14 consecutive days where the number of new cases of COVID has been reduced. The issue with this is with social distancing, the mathematical projections are quite unpredictable, and there's really a very limited methodology that we have right now to truly predict what this curve will look like. So every day what's happening at the Illinois Department of Public Health, if you look at this graph, it shows all the new coronavirus cases up till yesterday, actually. Every day, the Department of Public Health looks at this graph and tries to understand how the trends are changing and how the mathematical models need to account for these changes in order to predict when we can stop the shelter in place. So you can see that this data is a lot more complex than the graphs that are purely generated from a mathematical model. That's down below on the left. But they are looking for 14 consecutive days with decreases in the number of new cases. And what that will say is that the transmission is definitely going down. So we want to make sure that we're at the peak when we end the shelter in place so that we don't see the resurgence of cases as you can see in the blue line here. Okay. So now I want to shift a little bit about vaccine development. There's been a lot of questions that I've heard about why vaccine development will take so long. Vaccine development is really important because it gives us a way to protect ourselves from the virus before we contract it, and regardless of whether social distancing is in place. So first, I'd like to just briefly mention the steps that go into vaccine development. The first is the development of the therapeutic in the lab. So with cells in a dish or animal models, scientists will develop a deactivated virus that what their goal is to get the immune system of our bodies to respond to this deactivated virus and understand how to fight it in the future. So once they've developed what they think is an effective methodology, the virus starts to go, the vaccine goes through toxicology testing. And what the aim of this is to make sure that if it's injected into you, there won't be any negative side effects from the vaccine interacting with your organs that cause any immune response that negatively affects you or causes any inflammation. So once that's established at a preliminary level, a company or a lab will submit to the FDA. Now the FDA, their main job is to make sure that everyone that receives this virus is safe. When you're injecting a batch of chemicals directly into our bodies, there's a lot of things that can go wrong. So there's quite strict guidelines that the FDA has to make sure that the vaccine will be safe. So typically this process can take 1.5 years alone, but the FDA has been quite efficient lately in rapidly approving different vaccine trials. So after submitting to the FDA, there's a series of clinical trials. The first, the company will evaluate safety and immune response, as I mentioned before. So you also need to keep in mind that during clinical trials, these studies are performed on humans. So volunteers have to be recruited and agree to actually give themselves to this trial for the sake of vaccine improvement. So the first step is to make sure that there's no adverse immune response. and that the vaccine is safe. And that's phase 1. After the company demonstrates that, they can move into phase 2 and 3, where they need to show that the vaccine actually works and keeps people from getting infected and that it's still safe. So in order to make sure that it's safe, you need quite an extended period of time. The average is usually about 18 months to make sure there's no negative long-term health effects that weren't originally identified. And then once all those steps are completed, a company can start thinking about how to manufacture this vaccine. Chemical and biological components are very complicated to maintain the integrity of in a manufacturing setting. I don't know if anyone's ever thought about this, but proteins are very sensitive, so maintaining that quality of the vaccine is really important, and it's also quite challenging. So this entire process is quite resource intensive and time intensive, and actually quite risky for a company, because vaccines might not, in this case, vaccines like the COVID might not actually be financially beneficial in the long run. So at this point, I will take a pause for questions on this.
Okay, we have a lot of questions about this. Leonard would like to know, going back to, we know that this virus came from bats, and you talked a little bit about, the transmission. Why did China decide to reopen their wet markets if they knew that this was a risk?
I am not really sure about the dynamic there and what the decision was. I think our FDA is quite stringent and does a really good job of trying to keep people safe, but other countries might not necessarily have the same infrastructure or have the same considerations of what's safe and what's not. I also think it's important to keep in mind that, as I mentioned, it's not very common for this to happen. It's kind of like a perfect storm of a lot of different things having to happen at once in order to have this virus mutate. In the past, there's been other coronavirus outbreaks, and even an environment as simple as a farm could cause a similar effect. So it's really hard for me to say there, but I think that it could happen in any environment, and it's quite unfortunate, but it's also like a 1 in a million chance of something like that happening.
Okay, so another question, we're waiting on this vaccine, and a lot of experts are saying it's going to be a year or more until we get it. But let's say that you are unfortunate enough to contract the virus. How long do you need to self-isolate, and particularly from the people who live in your home, like your kids or your spouse? Do you have to wait until your symptoms are gone? Do you have to wait till your symptoms are gone plus a few days? What's the recommendation there?
I think if you do come down with the virus, your doctor, your primary care physician, will provide insight to that. I believe what I've read so far is that you have to no longer be infected with the virus. I know they're doing tests now in some areas for antibodies, and they're also doing tests to see if you actively have the virus, and at that point it's definitely safe, but I'm not exactly sure in terms of how they can tell when that stops if you don't get tested again, whether it's just if you no longer have symptoms. But I think if you are infected with the virus, a physician could provide some insight there.
Okay, and then if you do come down with the virus, you're healed, you're living your life, is it possible to get it again?
Yeah, that's a really good question. Right now, we don't know. So based on the types of antibodies that we are producing in response to the virus, there's 2 different types. There's a short-term antibody and a long-term antibody. We don't know how effective either of those are going to be at protecting us in the long term, which is why I mentioned companies don't know if this vaccine is going to be financially beneficial to them because they don't know if once everyone gets it, if they're going to need it again, or if it's going to be something like the flu where we need to be revaccinated yearly.
Okay, and for those of us who haven't taken a biology class since high school, can you remind us what an antibody is?
Yes, so an antibody is a type of protein that your immune system uses to fight off the virus. So there's a couple of different types and they all have different functions. There's some antibodies, now these are just like proteins that are produced when you first get the virus, and they kind of serve as a guide to your immune system on how to actually fight off the virus.
Okay, and while we are waiting for a vaccine, is there a drug treatment for this disease? How are they treating it?
Yeah, that's a really good question. That's a good transition to my next couple slides. So I'll just move on and stop there for now. But first I wanted to show all the companies that are actually developing vaccines and a little bit about the different types of vaccines. So there are whole virus vaccines where the entire virus is deactivated and packaged, but then there's also subunit vaccines where parts of the virus that are the most informative to the immune system are packaged and then administered. And then the last type of vaccine that is being worked on right now is called a nucleic acid vaccine. And in that type, DNA or RNA from the virus is directly injected into the immune system. So now I want to talk a little bit about drug development, leaning into Amy's questions. So the main goal of the current drugs that are being used is to stop the virus reproduction. And this can work in a couple of different ways, either from stopping the virus from infecting the human cell, or also there have been some drugs that reduce the viral load and help the lungs actually to breathe better. So it can either target the symptoms, or it can target the virus reproduction, or the drugs can target the virus reinfection of the human cells. So this chart here shows some drugs that are currently being tested against COVID-19. The interesting thing with drugs compared to vaccines is that drugs can be used for one application and then tested with another once they're proven safe. So whereas vaccines are very specific to one type of virus, like COVID in this case, drugs target the mechanism of COVID infection. And as long as they've already been proven safe by the FDA, they can be tested for this new application. Does that answer your question, Amy?
I think so. Can I ask a few more questions on this note? Okay. So going back to social distancing, Rachel would like to know if you are doing a really good job of self-isolating and your friend is doing a really good job of self-isolating, is it safe to spend time with them? Can you, spend time with people outside of your home if you are doing a good job of limiting your exposure?
I think it's safest not to, because you just don't know if someone's symptomatic or asymptomatic. So just because, like I mentioned before, about 40% of people could actually have it and just not be producing symptoms. If you had been self-isolating for a very extended time period, and there, like we mentioned earlier, if you're living in a bubble, if there's no chance that you've been exposed because you haven't seen anyone and there's been no one coming in your house and it's been a couple months, then potentially. But I think right now we haven't been social distancing or sheltering in place long enough for you to know for sure that you are asymptomatic or not. So there's still a very good chance that you yourself have COVID and then you could transfer it to your friend.
Okay. And what about You see your neighbor walking across the sidewalk and you go out to your front door. You're more than 6 feet away and you just want to yell across your yard to your neighbor. Is that safe?
Yeah, I think that's fine as long as there's no chance of transmission. No exchange of hand touching or particles from your mouth or your nose. I think as long as you're not necessarily breathing in the same vicinity, that it's safe.
Okay. And going back to immunity and antibodies and your immune system, are there things that you can do to improve your immune system to make you be able to fight off this virus better? Are there foods that you could eat or other, home remedies that you can take to try to put yourself in a better place if you do get it?
Yeah, I haven't talked specifically about different foods and fruits, but healthy food, fruits and vegetables, eating well in general can definitely help you. Also, exercising and just keeping your body healthy is really beneficial. And with that in mind, also keeping stress levels down. I know there's a lot of uncertainty right now in all sorts of areas of our lives, so that can be really stressful. But the better you maintain your stress, the better your immune system will be able to fight off any infection. There's been a lot of studies that have shown your immune system is quite closely related to stress and anxiety. So just being less stressed in general and exercising and eating well will keep your immune system up.
Good to know, but not always easier said than done, I think sometimes, especially while we're all kind of cooped up in the house right now. That is all the questions I have on this topic. I think we're ready to move on.
Awesome. Okay, so moving on from drug development, I wanted to talk a little bit more about antibodies and convalescent plasma treatments. Recently, this has become something we've heard a lot about in the news. Is COVID-19 patients who have recovered, they have very high concentrations of antibodies in their blood from successfully fighting off the virus. So their blood can then be processed and these antibodies isolated and injected into patients and also people who have not yet gotten the virus. So basically in that way, where you see the prophylaxis, what that just means is people's antibodies can be used as sort of a vaccine before we even have one to prevent healthcare workers in some areas that haven't seen the peak in the high numbers of patients yet, like we have, not even get the vaccine, not contract the virus because they've been injected with these antibodies. In some cases, the antibodies have also been effective as therapy. So there was one study that treated 5 different patients with these antibodies, and they saw significant improvements in their health, although this isn't very well understood yet. Only patients who have recovered with a very high level of antibodies are eligible to donate the plasma, but it is a way that we're exploring to continue to treat critically ill patients. This has been something that we used in 2014 with Ebola. So there is some precedent in seeing its success, and it's a promising way to try and stop healthcare workers from contracting the virus as frequently, but it still requires FDA approval. So only certain hospitals right now are actually able to use this as a treatment. There's a lot of push to getting this streamlined so that more people can use the antibodies as a pseudo vaccine before one's prepared, and try and stop healthcare workers from contracting, but also use this to treat critically ill patients. And then I'll also briefly talk about some other technology that's been really beneficial in helping us treat patients that have COVID. So one is ventilators. We've heard about the shortage of ventilators, and that's a big problem. The purpose of a ventilator is to push air into the patient's lungs so that it supports the expansion and contraction of the breathing muscles. And when we get really sick, it can be difficult to take deep breaths. So the ventilator is necessary to support the breathing process. We also talked about masks earlier. Self-sanitizing masks are something that are currently being developed in research. And the way this works is there's virus neutralizing particles in the masks so that when healthcare professionals breathe in or out, there's no transmission of the virus. 3D printing has also been something we've used a lot during this during the virus outbreak to customize healthcare masks and also to use face shields. The masks that fit over your nose and mouth can be very uncomfortable for professionals to wear for over 8 hours at a time. So there have been some labs that are customizing 3D printed masks so that not only are they reusable, but they fit to the healthcare provider's face much more securely, much more comfortably. And then you can just use an interchangeable filter, and that stops virus particles a lot more effectively. And then we already mentioned this, the last thing is managing stress. So one of the best tips is to take a break from the news every day. I know it sounds hard and you need to stay informed about what's going on in the world, but it can become quite stressful and quite depressing to have the news playing on nonstop every day. So maybe an hour or 2 hours each day, just turn the news off and give yourself a mental break. Also keep your body healthy. I know it's pretty rainy right now, but as the nice weather approaches, try and take walks and plan some time to be outside, but also plan time for events that you really enjoy. And that being said, check in on your friends and family regularly. This is quite stressful time. I know we talked earlier about shouting at your neighbor across the yard, and that's totally okay to have conversations with people through glass or in any way that you're not. I think a good guideline is try not to be breathing the same air or in the same breathing vicinity. Some of the best informative resources that you can have, there's a lot of conflicting information right now, is medical professionals. Always listen to your doctor. But then also open source research journals can be really beneficial if you want to directly connect with what's going on in academic or industrial research labs. And also company websites are quite transparent if you're interested in drug or technology or vaccine development. I linked Johnson & Johnson here because I think their website is really clear on what their goals are with the vaccine that they're trying to develop. And the CDC COVID-19 website is also very informative with the face mask information. Like I had the diagram on how to craft a face mask from a t-shirt. So you can find a lot of really beneficial resources there. And with that, I will take any more questions if people have them, and I'm going to link my email here so that if anything else comes up, feel free to send me an email, or if you're interested in any of the other topics that I talked about, please don't hesitate to reach out.
So for your last slide, I just need to plug the library and remind everybody that we are your information resource in Glen Ellyn. Even if you don't live in Glen Ellyn, you can contact us and we will help you navigate this. There is so much information right now, and it's really, really difficult to know what your good sources are. And this is our bread and butter. This is what we do. So if you are having trouble navigating information, you have questions about this, you don't know if a source is accurate, you need, more information and you're not finding it, don't forget that you can contact us. We are not quite open for business yet, but we are here for you. You can contact us on social media. You can also email reference@gepl.org and we will help you. Just doing my due diligence as a librarian to remind you that the library is here to help too. So I've had a couple of questions come in. Can you talk about the different systems in your body that COVID is affecting? We know that it's affecting the lungs and the respiratory system, but is it also affecting the neurological system? Is it affecting, your digestive tract? Can you talk a little bit about that?
Yes. So I want to precurse by saying that there's a lot that we don't understand right now, and it's really hard to start a lot of studies with. Right now, our focus is on preserving safety as much as we possibly can. The number one body system that COVID has been attacking is respiratory. And that's because of the specific, if you remember, I had that diagram of all the different proteins and mutations that happened between the bat to other animal to human transition. There's a lot of very specific mutations that happened. And so within those mutations, what happened is it developed a specific affinity for what we think right now is lung cells. We haven't, that I've seen, necessarily understood any other areas that the virus attacks besides the lining of our lungs. With that being said, don't underestimate the importance of the lungs. So a lot of side effects that we've been seeing in influences that are in other systems are primarily caused from the virus's presence in the lungs. The lungs interact with the kidneys in ways that we don't traditionally think about, and also when the virus does attack the lungs, that compromises the oxygen content throughout the rest of our body. So it's a very complicated issue, and I think that when the lungs are involved, all our body systems are interconnected, so it's hard to say what the virus directly affects. But right now, the effect of the virus on the lungs does cause issues with the digestive system, the metabolic system, and I haven't heard much about the neuro system or the cardiovascular system yet, but I just keep in mind that it's rapidly evolving.
Okay, and so if we can talk a little bit about testing, I know that there's been a lot of talk about rapid tests and different types of tests. Why are there so many different types of tests, and why do some of them take longer than others to come in?
Yeah, that's a really good question. So if you remember, the way that we test for the virus right now is by looking for little fragments of the virus DNA. So I'll start the first part of the answer to that question is there are so many different tests because there's a lot of genetic material that the virus has. So what's happening is a lot of the different tests look at different parts of the virus's DNA. So each test has a different snippet of code and is trying to identify that within a person. Now, they take a very long time just because that process is so complicated. We don't understand a lot about specific proteins associated with the virus that the virus produces. Typically, when we get infected with a virus, it makes us do its bidding, and it makes us, our cells, execute all the functions that it needs. So when we're infected with the flu, we understand the flu very well, and we can look at specific protein or another specific molecule that's associated with the flu, but we don't understand COVID at that level yet. So right now, the only way that we have to test for it is by looking for specific sequences of its DNA. So that's really why the testing takes so long, is because that process is so complicated, and it's really the best way we understand right now to test for it.
Okay, and are those test results reliable? Are like the rapid tests, are they more reliable than the ones that take a little bit longer?
Yes, so I think the rapid tests are just as reliable as long as the sample is collected correctly. There's a very, very low chance of a false positive. However, there is some risk to a false negative just because DNA and genetic material in general is very sensitive. So if it's not collected correctly, that Lego piece that we're trying to match won't be there anymore, or it could be degraded, and then there's nothing for us to match, and the system will fail because the sample wasn't collected correctly, and there was some degradation that happened, and now we don't see it. So in general, they're all quite accurate, but there is some small risk of a false negative at this point.
So we are hearing a lot that COVID is pretty similar in genetic sequence to SARS and MERS. Is that correct?
Yes.
And, you know, drawing on our experience from those outbreaks, does that help us understand COVID? Is the testing similar? Can we reuse those tests?
Yeah, it absolutely is similar. A lot of the drugs that we're using right now served as the foundation for that. I think the tests that that we're using now were derived from that experience. Unfortunately, SARS research and MERS research was kind of cut down. So hopefully one positive thing that comes out from this is that after we make it through this virus outbreak, we don't cut off all the research on treatments and vaccines for COVID-19 because we are going through a similar issue right now to where we cut off all the research for SARS and MERS, and now we're kind of wishing that if that research had kept going, we would be more prepared now to handle COVID, but I don't think we understood those viruses as well as we could have, and now we don't necessarily have that information to leverage with COVID.
That's an interesting perspective. So when can we expect more widespread testing? Are we waiting on manufacturing delays? Is there an estimate as to when, you know, in Illinois, we're reaching about 10,000 tests a day, but that's clearly not enough. So what's the holdup with testing?
Yeah, I haven't seen any predictions on different how we can further increase it. The method that we're using right now requires a lot of special equipment that a lot of labs don't have. And it takes about 2 to 3 hours to run a single test. So until we can understand this virus better and maybe think of a better way to identify it as being infected, there's not a good way for us to improve the tests. I've noticed that a lot of people are pushing towards antibody detection and trying to understand if maybe we can look at a person's antibodies to see if their body is actively fighting off this virus. And then I've also seen some work on chest CT scans, but I don't know that either of those methods will be widespread enough, fast enough to actually improve our efficacy. Right now, there's a lot of small improvements that are being made gradually, and that's kind of having a domino effect where with each small improvement, with each lab that equips itself to perform the tests, we get improved testing capabilities. So until we can really understand a better marker, I'm not sure that there's a clear path for more widespread testing.
Okay, so we had a blood drive earlier this week at the library, and we are hearing about a lot of blood drives in the community. First of all, is it safe to donate blood?
Yes, the Red Cross, and I'm sure that all the blood drives are being very precautious about social distancing matters and disinfecting. As I mentioned, disinfecting makes everything much safer, and it's absolutely safe to donate blood right now.
And we talked a little bit about convalescent donations of plasma, but if you haven't had it, what are some reasons to donate blood? Is it being used for COVID patients?
Yeah, it's not necessarily being used right now for COVID patients, but there is a shortage because of a misconception that it might not be safe to donate blood, and a lot of people are working to social distance. So that just makes a lot less donors and a lot more people who necessarily normally would donate a little bit more hesitant to go out and donate. But it's really important to still have blood because there's a lot of different reasons that physicians use that blood, and they don't necessarily use it to treat COVID patients, but they still need it to treat other incidents, that other medical problems that people are still experiencing, like injuries from car accidents or other issues that people might require a blood transfusion. So it is still really important to keep up our blood banks.
Great. So let's say you are unfortunate enough to contract the virus and you are so sick that you go on a ventilator. How many people who end up on ventilators survive? Is that, we know it's an effective treatment, but is that kind of like the end of the end, or is it a life-saving treatment? I mean, can you talk a little bit about how ventilators play a role and what they do to keep people alive while they're recovering from this?
Yeah, so the issue with ventilators is they're intended to support your lungs, your lung muscles, but they kind of take the load off your lung muscles, which can make them be even weaker. There has been some statistics that have shown that when you start on the ventilator, it becomes a lot harder to come off. I think about 30% of people get off of the ventilators. Now, keep in mind that they're absolutely used as a last resort to start with, and patients are typically in critical condition. A lot of doctors now are looking into alternative methods where there's different other breathing support techniques that they're trying in the hospitals to try and avoid putting patients on ventilators. But it does make it a lot harder to come off, just because it's really hard to re-strengthen your lung muscles once the ventilator has been supporting your breathing and they haven't had to work independently for an extended period of time.
Okay, so hearing you talk about this, is it safe to assume that we just don't have enough information to know whether we're safe overall, even if we're taking the proper precautions?
Yeah, so I think that you can assume that if you're being precautious in disinfecting surfaces, maintaining social distance, not interacting with people unless you have to, wearing masks when you go to the grocery store, washing your hands frequently, if you're taking all the necessary precautions, you're most likely going to be safe. A lot of the people who are getting sick are still going to work and still having high interactions with a lot of other people, or not necessarily social distancing. But I think as long as you are following everything and disinfecting surfaces, washing your hands, that you can assume that you're safe. I think that you might get yourself very stressed, so it's better to make sure that you're recognizing that you are safe, especially if you're doing the social distancing. And becoming stressed might actually be worse for your immune system, like we talked about. That's a good way to try and not get sick, is by maintaining a good level of calm, even though, like we said, easier said than done.
Great. And I have one last question before we wrap up. If our attendees today remember one thing from this presentation, what do you hope that they take away from it?
That's a really good question. I want them, I hope you guys all realize that you have some sort of peace of mind from all this, that there's a lot being done to fight COVID. We have a lot of drugs that are being tested, and the convalescent plasma therapy is quite promising. And then in accordance with the last question, if you are following all the guidelines, you will be safe. If you're disinfecting all the surfaces, the virus doesn't really have a chance. If you're washing your hands, there's no need to become overburdened with stress, because there's a lot of positive things that we're headed in the right direction for, with the vaccines and drugs, and social distancing is really doing a lot to protect us.
Okay, well, on that note, we are just about 10 minutes over time. I just want to extend a very sincere thank you to you, Rebecca. Thank you so much for sharing your expertise and your knowledge and your time. I would ask for an applause, but it's only going to be from me. So I really thank you. I hope that when we reopen, you come in and see us. We'd love to talk to you. We'd love to get to know you. And I just want to remind everybody that Rebecca is one of us. She's in Glen Ellyn, and she's a community member, and she's doing all the same things that everybody else is to try to protect herself from the virus, right?
Thanks so much, everyone, for coming today.
Thank you.